Hi students, this is your second video lecture of Module 5. Last time we were discussing taxonomy, which is the classification and naming of living things and how that follows phylogeny, the evolutionary history. In this video lecture, we're going to be focusing on organisms, the prokaryotes, which include both organisms in the domain bacteria and archaea. So prokaryotes, just realize, isn't a domain, it's a category of living organisms. And these are those single-celled organisms that are really simple, in both bacteria and archaea are prokaryotes. So this is that phylogenetic tree, the tree of the three domains. Okay, so the prokaryotes include bacteria and archaea. So if you look at this evolutionary tree here, we're going to be talking about bacteria and archaea. They're both prokaryotes. We're not talking right now about eukaryotes. All right. One thing that you can tell by looking at this tree is that archaea and bacteria have this common ancestor, this branch point. Right? Archaea is more closely related to eukaryote because they have a more recent common ancestor than bacteria and eukaryote. You'd have to go all the way back here. So I'll be telling you that archaea and eukaryote are more similar than bacteria and eukaryotes. All right, so just as a reference point, size of these prokaryotic organisms. So they are very tiny. So these were thought of to be the first organisms on Earth, right? They were more primitive than our cells. And this picture kind of gives you a reference point of an animal cell, right? Which is eukaryotic, right? It has a membrane-bound nucleus. They can't even show you the whole animal cell here, but look at the size of the bacterium, which is a type of prokaryote, much smaller. And even smaller yet are viruses that we're not going to really talk about. Viruses are none of the domains because they're not considered living because they can't reproduce on their own. And remember, one of the criteria for life is that it has to be able to reproduce on its own. But viruses are these tiny, we call them genes in a box because they're just genetic material in a protein coat. They don't have ribosomes to make their own protein, so we don't classify them as living. But this just gives you the idea of the size. Prokaryotes are everywhere. They're in the air, they're in the water, they're in our bodies, outside and inside our intestine, on most of the objects that we touch, but we cannot see them. The structure of a prokaryote, remember, we've talked about this already. They don't have a membrane-bound nucleus, right? We call it, their DNA is just right here in this region that we refer to as a nucleoid region. They also don't have other membrane-bound organelles, like they don't have endoplasmic reticulum or a Golgi apparatus. They have an outer cell wall, right out here, the cell wall, and then a membrane and within the cell wall level underneath it. Many move by flagella. All right, and sometimes they have extra little genetic material called plasmids that are basically, I'll draw one here, just an extra little beneficial genetic material. This is a lot of times in bacteria where antibiotic resistant genes are found, and these plasmids can swap between bacteria. They can exchange genetic material that way, and that's in addition to this main central chromosome. Prokaryotes reproduce asexually. When you put A in front of a word, it means without, so without sex. They have one parent, and they duplicate into two identical prokaryotic cells. That is referred to as binary fission. Okay, so here you see one bacteria splitting into two. It's kind of like a simplified mitosis. They do have to replicate their genetic material, but because they don't have a nucleus that's membrane bound, we don't call it mitosis, but it's like a simplified mitosis. One cell becomes two, they're both genetically identical. Now, bacteria can reproduce every like 20 minutes or so. And every time they reproduce, they have to duplicate their genetic material. And every time you duplicate the genetic material, a mistake can happen, right? If you're trying to copy ATGC, sometimes accidentally it'll copy AGCG, right? So that's referred to as a mutation. A mutation is a change in the genetic sequence. And sometimes those mutations actually for bacteria can be beneficial. That's how they evolve and adapt to their environment, because they don't reproduce sexually, so they can't get genetic variation by sexual reproduction. And here's just a closer up picture of that. So this is just a picture showing you how quickly bacteria population can grow. So for example, E. coli, which is the bacteria found in our guts, it's also bacteria that can make us sick. It's found in fecal matter. So bacteria, E. coli, can undergo binary fission every 20 to 30 minutes. So you can see within an hour, the population can grow. And this is referred to as exponential growth, right? And that's why bacterial infections can happen really quickly. For example, food poisoning, right? What is food poisoning? Generally, it's because food that you've eaten is contaminated with some sort of bacteria, such as E. coli, and you eat it, and within a few hours, that population has grown exponentially and can make you feel sick. Some bacteria can also produce these structures called endospores. And an endospore is like a protective covering within its genetic material, so that if conditions are unfavorable, like there isn't enough water, the temperatures are too high, there's toxins, the rest of the bacteria here will break down, but the endospore will exist. And then when conditions become favorable again, the endospore can produce the bacterium again. This is actually a problem in hospitals because endospores sometimes can withstand the sterilization processes that hospitals use, right? But for them, it's an advantage to survive. And this is just pictures of bacteria, and these little blue structures are the endospores. Bacteria, now we're talking about the prokaryotic bacteria. Remember, prokaryotes consist of bacteria and archaea. Bacteria are actually the more common of the two. There's 9,000 or so bacteria have been named. There's probably many more that exist, but those are the ones that have been named. So then we have all these ways to categorize them. And one reason it's really important to differentiate them and categorize them is in medicine. That's how we know which bacteria is causing someone to be sick, and therefore which antibiotics should be used. So one of the things that's often used is gram staining. So bacteria have cell walls that have this molecule called peptidoglycan, which is like a carbohydrate protein mix. And we stain them in a lab to see which category of cell wall they have. So we group them into either gram positive or gram negative. Gram positive appear purple because their cell wall keeps the dye. Gram negative do not keep the dye, so they appear more pink. And so if it's gram positive or gram negative, that's one way to know what antibiotics should we use. Another way that we classify bacteria is by just their overall structure, all right? And so the shape, right, can be either the spiral shape. There are many STDs like syphilis that are the spiral shape. We call these spirili or spirochetes. They can have this rod shape like the one in the middle. E. coli is an example of a rod-shaped one. Or they can have this cocci or round shape. A lot of skin bacteria like staph bacteria is this round shape. Bacteria can have, one of the things about bacteria is because they're such ancient organisms, they've been around way longer than humans. They're the most ancient of all of life. They've evolved in different locations on Earth. So they've evolved ways to survive with oxygen, without oxygen, either with or without. So their metabolism, right, the reactions in their body, we can classify bacteria as being either obligate aerobes. Obligate, think they're obligated to have air. So these are ones that use oxygen, so they're unable to grow without oxygen. Then there's obligate anaerobes. So these are ones that are obligated to use. Remember, if you have A in front is without air. So they're unable to grow in the presence of oxygen. So these obligate anaerobes don't use oxygen. And then there's facultative anaerobes, which means they can do either. They can grow with or without oxygen. So even though bacteria seem very simple, their cells are very simple, they're actually very successful at life, right? They can grow in diverse environments. Bacteria are mostly heterotrophic. So what do we mean by heterotrophs? Heterotrophs are the ones that consume organic material for their energy. We talked about this when we looked at cellular respiration versus photosynthesis. So heterotrophs, right, can't use the sun energy to make their organic molecules. They have to consume them. So most bacteria are heterotrophs, right? And even most prokaryotes are heterotrophs. The exception in the bacteria are a group called cyanobacteria. These are photosynthetic, so they're autos. They can produce their own own food. Cyanobacteria were also the ones that are thought to have been the first organisms to put oxygen into our atmosphere. There are also bacteria that are decomposers. This is a very important part of what bacteria do for the earth. They break down dead or decaying matter. If there weren't bacteria, there'd be miles of dead stuff just on Earth because there'd be nothing to break them down. And by breaking down dead matter, they recycle all of those elements, right? The carbon, the hydrogen, the oxygen. Bacteria can be free living or symbiotic. Remember, symbiotic is a close relationship with another organism. Bacteria can either be commensalisms when the one benefits, the other one doesn't. So you don't have to memorize these terms, but they're all just different symbiotic relationships. So we have a lot of bacteria living on our skin that doesn't affect us, but they benefit because they have a place to live. They can be mutualistic, where both benefit. We have bacteria in our gut, right, that helps digest our food and in the process produces vitamins that we then absorb. So that would be an example of a mutualistic relationship. We both benefit. And then there's the parasitic relationship. That's the one where one benefits, the other is harmed. And so those are the bacteria that cause us to get sick, that have more of a parasitic relationship. I would say that bacteria kind of have a bad rap. I would say that there are many benefits to bacteria, but there are those that cause us to get sick. So here are some types of illnesses that are because of bacteria. So STDs are because of bacteria. Strep throat is by Streptococcus bacteria. Certain types of pneumonia, right? Skin, people can get staph infections because of the bacteria on our skin. We can have GI-like illnesses where you have diarrhea. Often that's because of bacteria. And so Lyme's is one that's pretty common here in Wisconsin, and that's because of bacteria that ticks have in them. It isn't the tick that's getting you sick, it's the bacteria within the tick that gets in your body and gets you sick. I did talk about the cyanobacteria. These are the one type that are photosynthetic versus, right, heterotrophs, which is what the other ones are. And they're believed to be the first organism to release oxygen into our atmosphere. So the primitive Earth did not have oxygen. After the cyanobacteria evolved, they released oxygen. All right, and lastly, the prokaryotes that are the archaea. Remember, archaea is a domain that's more closely related to eukaryotes than bacteria are to eukaryotes. Initially, archaea were just considered bacteria, but then a scientist, Carl Woese, identified that a portion of the genetic material on the archaea showed that by base pairing, right, that they were actually different than bacteria and more closely related to eukaryotes. Archaea do not have peptidoglycan in their cell walls like bacteria do. So biochemically, they're more like eukaryotes than bacteria are, but they're still prokaryotic cells, so they're still a prokaryote. They live in extreme environments. Sometimes we call them extremophiles. They love the extreme environments, like these salty, this is the Great Salt Lake of Utah, where nothing lives, but these prokaryotes have a way to survive in these environments. We call these halophiles, the ones that love the salt. They're ones that live in hot thermal vents. We call those thermophiles. They love these hot environments where nothing else lives. Other ones live in these swamps where there's large amounts of methane. They can survive in there. Okay, as a summary, prokaryotes are these cells, single-celled organisms. There's no multicellular prokaryotes. They have no nucleus, no organelles. Bacteria and archaea are both types. We categorize bacteria by gram-positive or gram-negative. We categorize them by the shape. They can be growing with or without oxygen, right? That's the obligate aerobes, facultative anaerobes, or the, right? Some cause disease, but some are beneficial to us. And then the archaea differ from bacteria because of their biochemistry, so that's why we don't put them in the same domain. They are extreme organisms. They're found in extreme environments, like the hot springs and salt lakes and swamps. All right, take care, everyone.